So in this video, we are going to see about epidermopoiesis, that is epidermal dynamics and kinetics. So this epidermis is under a phase of constant proliferation and differentiation. So for this to normally happen, the epidermal cell production should match the rate of cell loss. So the cell cycle, it consists of 4 phases. So the M phase or mitotic phase, G1 otherwise called as the interphase or intermitotic phase or postmitotic phase, S phase which is the stage of active DNA synthesis, and then G2 it is nothing but the resting phase. There is another phase called as G0 which is called as quiescent phase. So these stem cells are nothing but they are small clusters in the basal interfollicular epidermis. So if you see the cell cycle, the intermitotic time is the time interval between two successive mitoses. So the cell cycle happens during the intermitotic time, that is a time interval between two successive mitoses, and the 4 phases as I mentioned already, M, G1, S, G2, and the 5th one is G0 otherwise called as quiescent phase. So the duration of the normal cell cycle is around 50 to 457 hours, whereas in psoriasis it is only around 36 hours. And the mitotic division, it takes place around every 18 to 19 days. So it takes place in the basal layer of keratinocytes. So what is epidermal turnover time? It is the time taken by keratinocytes to pass from basal layer to stratum corneum and the skin surface. So the approximate epidermal turnover time is around 52 to 75 days. That is, from basal layer to stratum corneum, it takes about 12 to 19 days, and from stratum corneum, that is through the stratum corneum, it is about 14 days. So the regulation of epidermopoiesis, here we have stimulatory signals, inhibitory signals, and then the signal transduction complex, etc. We will see one by one. So the key components mainly include NF-kappa beta, WNT beta catenin pathway, alpha catenin, sonic hedgehog pathway, this P63 which is the epithelial immunohistochemistry marker, and the beta 1 integrin. So the stimulatory signals, it mainly includes growth factors like epidermal growth factor, transforming growth factor, and then this you have autocrine and paracrine stimulatory signals here. So this TGF alpha, it has an autocrine action. It binds to epidermal growth factor receptor and stimulates the growth of keratinocytes. Another one is amphiregulin. It is also an autocrine KGF stimulator. So next one is interleukin 1, 6 and granulocyte monocyte colony stimulating factor. And then here you see next one is the paracrine factors for keratinocyte growth stimulation, which includes the dermal fibroblast and then the endothelial cells. So other growth factors are fibroblast growth factors. So all these growth factors and then this platelet derived growth factor, interleukin 1 beta, TNF, all of them finally induce the keratinocyte growth factor gene. So which is highly specific for keratinocytes. So these are the stimulatory signals, and then this inhibitory signals, it mainly acts by negative feedback mechanism and includes this interferon alpha, gamma, TNF alpha, and then this TGF alpha, etc. And this apoptosis, it is a major homeostatic mechanism both in skin and also in hair follicle. In hair follicle, this apoptosis marks the onset of catagen phase. So next is the signal transduction pathway. It happens mainly through hormone receptor complex and then growth factors as I mentioned already, and then protein kinase C, inositol phosphate, and protein tyrosine kinase, etc. So these integrins, they are mostly present in the basal layer, and it has a bidirectional communication. That is, it acts a bidirectional communication between the matrix molecules like collagen, fibronectin, and laminin, and the cytoskeleton of keratinocytes, and depending upon the pH, amount of calcium, and gene expression, it helps in the proliferation and differentiation of epidermal cells. So other factors include low calcium, which inhibits the differentiation but stimulates proliferation, whereas high calcium, it inhibits the proliferation. And vitamin A and retinoic acid, they are needed for the normal morphogenesis and differentiation of epidermis. So this epidermal differentiation, During differentiation, the cells move from basal layer to skin surface and then they undergo terminal differentiation to form the stratum corneum. So this process involves 5 steps. First one is keratinization. Keratinization is nothing but formation of keratin within the keratinocytes. So this takes place with the help of filaggrin, that is filament aggregating protein. Number 2 is cornified envelope and then intracellular lipid formation and then membrane glycoproteins and then growth factor receptors, adhesion proteins and blood group antigens. So all this formation involves complete process of epidermal differentiation. Next one is the formation of cornified envelope. So in this it involves formation of envelope precursors like involucrin and then the cytosolic precursors like SPR, SPR1 or pancornolin and other envelope proteins including scalp or elafin and keratolinin or cystatin. They are cross-linked by transglutaminase and the membrane associated precursors like envoplakin, periplakin and other major component including loricrin, which is a cysteine rich protein. So this cornified envelope, how does it look like? Okay. So this is the stratum corneum cornified envelope. Okay, these are the lipids in the stratum corneum. This is the involucrin. So here we have, these are the loricrin. So in loricrin, in between we also have this small protein rich protein. It's nothing but the SPR proteins. So here these are the keratin intermediate filaments which are linked together by filaggrin. So this is how the cornified envelope in stratum corneum looks like. So this loricrin, it depends upon calcium amount and in the keratinocyte differentiation, and also it is down regulated by retinoids. Cross-linking between plakin and involucrin and the desmosomal proteins and also the proteins and lipids. So all these cross-linking leads to a formation of scaffold which plays an important role in water permeability and barrier function of the skin. So next is the keratinocyte integrins. So this integrin, as we mentioned already, it forms a bidirectional communication and it also mediates the intercellular adhesions. So during differentiation there will be loss of integrin expression. So how does it helps us? It allows the keratinocytes to lose the adhesion to the extracellular matrix and help in the differentiation of keratinocytes. So next is the intercellular junctions. These are the junctions that link the adjacent keratinocytes. They help in mechanical, biochemical and also signaling interactions. So first one, there are 4 types. The first one is the desmosomes. So what this desmosome does is it connects the cell to cell and also tonofilaments to the plasma membrane. So these are the desmosomes. This, you can see the desmosomal plug here. There are two types actually. One is inner plug and another one is outer plug. They are two dense desmosomal plugs and they are about 15 nanometer in diameter. So this is the mid-layered zone in between the two plugs of the desmosome. It is about 30 nanometer in diameter and the desmosome, it contains desmosomal cadenins, that is calcium dependent cell adhesion proteins, which are, this is desmocolin and this is desmoglins. So this desmoglin and desmocolin both are desmosomal cadenin proteins. So these are attached to the keratin intermediate filaments with the help of desmoplakin, plakoglobulin and pacoflin, etc. So here you can see in the core we have calcium binding proteins, as I mentioned already, that is desmoglin and desmocolin. So this desmoglin 1 to 3 and desmocolin 1 to 3, they are specific for the epidermis. So desmoglin 1 and desmocolin 1, they are more in upper epidermis, whereas desmoglin 3 and desmocolin 3, they are more in the basal keratinocytes. So this is desmosomes. Number 2, we have adherence junction. They are calcium dependent and they are associated with actin skeleton through alpha catenin. Third one is gap junctions. So their component include connexons. They help in transfer of ions and small molecules between two cells. Number 4, we have tight junctions, and these are the major regulators of skin permeability and play an important role in barrier function of the skin. And the components include occludin junction adhesion molecules. So these are the intercellular junctions. We have the tight junction, that is claudin, occludin and junction adhesion molecules, and the adherence junction, which includes cadherin adhesion receptors and cytoplasmic proteins, and the gap junction connexons and the desmosomes we see already. So the applied aspects of epidermopoiesis is, if there is any abnormality in the epidermal development like somatic mosaicism or post-zygotic mutation, it can lead to aplasia cutis, and the mutation in genes for steroid or lipid metabolism can cause X-linked recessive ichthyosis. If there is any failure for the cornified cells to slough, then it can cause retention hyperkeratosis, and antibodies to desmoglein proteins can lead to pemphigus, and absence of filaggrin, it can also lead to harlequin ichthyosis, and keratinocyte abnormalities seen in conditions like psoriasis, eczema, graft versus host disease, pemphigus, etc. And the keratinocyte culture, it is mainly used in skin grafting in burns. Autograft is used in case of transplantation, and allograft it is used in biological things. And the keratinocyte, it is mainly play an important role in skin immune system called as skin associated lymphoid tissue. And psoriatic keratinocytes, the cell cycle duration I mentioned in the starting itself, it is around 36 hours. And the human growth hormone, when transferred to keratinocytes, they are used as gene therapy in epidermolysis bullosa. So the non-keratinocytes in epidermis are melanocytes and Merkel cells in the basal layer, and Langerhans cells in the suprabasal layers, mainly in the spinous layer.